ठीक है. सो हमने अभी तक फॉस्फोलिपिड का पूरा स्ट्रक्चर देख लिया, फ्लूइड मोज़ेक मॉडल देख लिया. नाउ मूविंग टुवर्ड्स ग्लाइकोप्रोटीन्स एंड ग्लाइकोलिपिड्स. तो ग्लाइकोप्रोटीन्स एंड ग्लाइकोलिपिड्स का एज सच क्वेश्चन नहीं आता, बट 4 मार्क्स का वंस अगेन इसके फंक्शंस आ सकते हैं. ठीक है. तो 4 मार्क्स के लिए हमारे पास 4 पॉइंटर्स लिखे हुए हैं. व्हाट डू वी एग्जैक्टली नीड टू राइट इन आवर स्क्रिप्ट्स. तो स्टार्टिंग ऑफ विद ग्लाइकोप्रोटीन्स. अच्छा, ग्लाइकोप्रोटीन्स एंड ग्लाइकोलिपिड्स आर एग्जैक्टली द सेम एक्सेप्ट कि प्रोटीन की जगह वहां लिपिड लग जाएगा. ठीक है, बट दे अपीयर एज अ सिमिलर प्रोजेक्शंस. ठीक है. So glycoproteins, they play a role in identifying the cells, which is important for immune responses. I mentioned this in the recording as well. Okay, there are two types of cells. One of them are the self cells. Self cells हमारे cells होते हैं. And then we have foreign cells. So whenever we are under the influence of any microorganism or bacterial attack or viral attack, so the cells of those are going to be known as foreign cells. ठीक है. So glycoproteins or glycolipids, they work as a highlighter in such a manner that they highlight all those cells that are foreign in nature, and they highlight and they do not highlight those cells that are not foreign, meaning that if they are self cells, they are not going to highlight them. Okay, so they act as a highlighter. Do not write this in your script that they act as a highlighter. This is just an example that they act like a highlighter and they highlight the foreign cells to trigger the immune response. Okay. Then glycoproteins act also like receptors for hormones and for neurotransmitters and other signaling molecules. I said that the molecules that bind with receptor proteins are also known as, other than the signaling molecules, what is their exact terminology? क्या कहते हैं signaling molecules? दूसरा नाम क्या था? उस्मान. याद करें. अभी बताया.
मिस
जी.
मिस याद नहीं.
अब्दुल रहमान.
Miss, can you repeat the question?
Yeah, I said that there's a good, better terminology for signaling molecules. What exactly is that? अभी बताया था पिछली स्लाइड में. सिग्नलिंग मॉलिक्यूल का दूसरा नाम क्या था?
Binding?
नहीं, लिगेंड्स.
Ligand.
अभी बताया नहीं था. देखा, मैं ऐसे क्विक चेक करती रहा करूंगी and then I'll come to know how attentive you are in the classes. I do appreciate that you are a part of live classes, but I will also appreciate the fact when you'll be attentive. ठीक है. ठीक है. चलिए, when these molecules bind, when these molecules, meaning when these ligands, they bind to glycoproteins, they trigger a response within the cell, leading to various cellular activities. This is one of the functions of glycoproteins. Then the other function is that glycoprotein they act like a glue stick. So meaning that they try to stick the cells with one another, and eventually they form tissues, because when multiple cells combine, they form tissues. ठीक है. So glycoproteins, they adhere the cells. They make these cells contact. They make these cells stick to one another. Then glycoproteins also contribute to the stability and flexibility of the cell membrane by interacting with other membranes. So these are core functions of glycoproteins or glycolipids. 4 marks के लिए आए तो write exactly the same things. कुछ miss out नहीं करिएगा, because if you miss out then you'll not get the marks. तो सारी चीजें जिसमें हाईलाइट करिए स्पेशली, they are the important keywords and they should be a part of your scripts. Okay. Now moving towards cell signaling. So slightly technical subtopic, but आसान सी बात है, हो जाएगी अगर मैं इसको ऐसे समझाऊं. If this is a cell, ठीक है, सेल के ऊपर एक रिसेप्टर प्रोटीन है, कहीं से एक न्यूरोट्रांसमीटर आता है, या एंजाइम आता है, या हार्मोन आता है. ठीक है. सो दीज मॉलिक्यूल्स आर नोन एज ligands. Exactly. When these molecules come, they bring certain sort of signal within themselves. Okay, and when they come, they bind with their receptor proteins. When they bind with their receptor proteins, this induces a signal transduction within the cell, and a signaling cascade then happens. Signaling cascade means that original signal is then amplified in order to achieve particular metabolic response. Yeah. So when that particular metabolic response is achieved, then that action happens within the cell. So cell signaling, basically in a nutshell, is a process in which certain sort of signal comes to the cell, it binds with the cell, induces certain signal transduction, and via signal amplification, certain metabolic responses achieved. Okay. So cells produce and release signaling molecules known as ligands. These ligands could be anything. It could be hormones, neurotransmitters, or enzymes. And ligands are often secreted from cells via exocytosis. So exocytosis abhi main aapko batati hu, because it will come in the later slides. They're often secreted from cells via exocytosis, where vesicles containing the ligands fuse with the cell's membrane and release their contents into extracellular space. ये वाला part, if you're unable to understand right now, कोई बात नहीं, because exocytosis का पूरा subtopic we do have. ठीक है? Then ligands can diffuse through the extracellular fluid to reach nearby target cells in the case of local signaling. And in the case of endocrine signaling, ligands such as hormones are transported through the bloodstream. तो जो hormones होते हैं, they are transported via bloodstream. While अगर कोई molecule है और within the cells उसकी जो है वो release हो रही है, तो वो nearby target cells पर जो है वो जल्दी से उसकी local signaling हो जाती है. But if something like hormone has to come, then it has to be transported via bloodstream. ठीक है? So these are very basic things that you need to remember for cell signaling. कि आपका signal transduce होता है when it binds to the receptor protein, original signal amplify होता है, जिसकी वजह से आपका पूरा जो भी cellular metabolic response होना होता है वो achieve हो रहा होता है. अच्छा जी, ये पूरे steps उसके लिखे हुए हैं. That ligands bind specifically to receptor proteins. This is exactly what I have just mentioned. इसको बस side से थोड़ा और देख लेते हैं. Ligands bind specifically to the receptor proteins on the surface of target cells. These receptors are usually highly specific to particular ligands, less like enzyme-substrate complex. Enzyme or substrate used to be a complementary shapes, right? उसी तरीके से ligands or receptors are of specific shape. And upon binding, the receptor undergoes a conformational change, which initiates the signaling cascade inside the target cell, and this process can activate various intracellular pathways leading to specific cellular response. अब जो भी सेल का रिस्पॉन्स होना है, अगर कोई हार्मोन आ रहा है, उसने कहा कि भाई ग्लूकोज के लेवल को कम करना है, तो वो अचीव हो जाएगा. दिस इज जस्ट एन एग्जाम्पल. कोई भी सेलुलर रिस्पॉन्स हो सकता है. ये हो सकता है कि कोई होम्योस्टेसिस का रिस्पॉन्स हो सकता है, कोई प्रोटीन को कन्वर्जन का हो सकता है कि प्रोटीन को अमाइनो एसिड में कन्वर्ट करना है या अमाइनो एसिड को प्रोटीन्स में लेकर जाना है. तो इट कुड बी एनी सेलुलर रिस्पॉन्स. ठीक है? And the activated receptor typically triggers a series of intracellular events, often involving secondary messengers. And ये बहुत सारे messengers involved in this entire cell signaling process, and they amplify the signal and direct it towards a specific cellular responses. Here are the movements. Movements are very easy. First one, diffusion. We're already aware with diffusion, it's the movement of ions and molecules from a region of higher concentration to a region of lower concentration down the concentration gradient. And uske factors you need to remember. This may come for 4 marks here. What influences diffusion? तो steepness, जितना ज़्यादा steep होगा concentration gradient, उतनी ज़्यादा diffusion होती है. Then higher the temperature, higher will be the diffusion rate. Then जितना ज़्यादा surface area होता है, उतना ज़्यादा increased rate of diffusion होता है. And as the distance over which diffusion must occur increases, the diffusion rate decreases. So you need to remember this, कि जब distance ज़्यादा हो जाएगा, तो diffusion का rate will decrease. And molecules like fats, which are small and non-polar, diffuse more easily across the cell membrane because they're soluble in phospholipid tails. ठीक है? So this is very important. This is something which is new. Okay, molecules like fat, which are small and non-polar, they diffuse more easily because it is easier for them to pass that hydrophobic pore. Hydrophobic pore is made by fatty acid की tails. ठीक है? So molecules जो छोटे होते हैं उनका diffuse होना is much more easier as compared to वो molecules which are larger in size. Then facilitated diffusion. So facilitated diffusion is a new a new addition in the movements, and it's new for you as well because A levels ka part hai. This was not the part of your O levels. So facilitated diffusion is a similar type of diffusion. Like it's similar to simple diffusion. It's passive in nature. It does not require energy, but now in facilitated diffusions, we'll have our integral proteins be a part of it. So channel proteins basically are assisting in facilitated diffusion, and they're allowing those ions or molecules which cannot pass through simple diffusion. So they provide the proteins provide a pathway, and with that pathway, those molecules are able to pass through. And this does not require energy. And molecules which are slightly big and they're polar in nature, they are allowed to pass via channel proteins. Okay, so facilitated diffusion mein channel proteins are going to play their part in order for bigger and for polar molecules so they could pass through the cell surface membrane. Okay. Then osmosis is similar to what you have studied in your O levels, that it's the movement of water molecules from a region of, you'll not write concentration, you'll write higher water potential to lower water potential through a partially permeable membrane. And you need to remember this word, that water potential means that it is the tendency of water to move out of a solution. And you need to remember that water potential is represented by this Greek letter, and H is or that water potential becomes negative when the solute concentration is very high. So this should be very obvious कि जितना ज़्यादा अगर पानी है उसमें जितना ज़्यादा salt डालेंगे तो water potential is going to be negative because आपने solute concentration ज़्यादा कर दी and you have reduced the water potential. ठीक है, so जितना कम water potential होगा उतना negative आपकी value हो जाएगी. All right, then ठीक है, this is the movement. ये भी आप लोगों ने O levels में किया, but recap के you can only use these words isotonic, hypotonic, and hypertonic when we are talking about the plant cell. Okay, do not use these words when you're talking about a red blood cell or any animal cell. You cannot use isotonic, you cannot use hypotonic or hypertonic. So when you place a plant cell in isotonic में से same potentials होते हैं. So जब potential same होते हैं तो उस पे cell के ऊपर there's no effect. But when you place a plant cell in a hypotonic solution, the water moves inside the plant cell and the plant cell will swell. But if you place a plant cell in a hypertonic solution, then water within the plant cell will move outside and your cell will shrink and it will eventually plasmolize. It will eventually plasmolize. Okay, but this is exactly the same case with red blood cells, other than the fact that you cannot use the word isotonic, hypertonic, and hypertonic, but rather when you place a red blood cell in a high water potential, it will burst because it does not have a cell wall to sustain the pressure. While if you place the red blood cell in a solution of low water potential, it will shrink. And then you have active transport. The active transport is the exact opposite of diffusion, that it's the movement of ions and molecules from a concentration of from a lower concentration to a higher concentration, and in this process you use ATP. Okay, then bulk transport, yeah, so this is new, and bulk transport is very important for you guys. So ये बिल्कुल new है, इसको बहुत carefully समझें. Bulk transport means that you are transporting something in a very large quantity. Okay, अभी तक जो हमने बात करी, it was about diffusion, it was about facilitated diffusion, osmosis, and active transport. And in that, we were just talking about ions and molecules and water molecules. We were not talking about large molecules. So basically, large molecules such as antibodies, they are transported via bulk transport. Okay, bulk transport means कोई चीज large amount में जब transport होती है उसका हम bulk transport कहते हैं. And this transport movement, this requires energy in form of ATP. Okay, because इसमें बहुत ज़्यादा volume of material involved हुआ होता है. So basically, bulk transport is of two types. I'll suggest that you make this flow chart in your notebooks. Bulk transport, BT means bulk transport. इसकी we have two categories. I do not have a page बना और अच्छे से बनाती, but इसको सब देखें. Bulk transport is of two types: endocytosis, please write this word, complete word, ठीक है. और हमारे पास we have exocytosis. Bulk transport is of two types: endocytosis and exocytosis. Exo means outside, कोई चीज बाहर जा रही होगी तो exocytosis से जाती है. और कोई चीज अंदर आती है तो वो एंडोसाइटोसिस से आती है, इनसाइड, ठीक है। दिस इज जस्ट फॉर योर ईज, नहीं बनाना तो इट्स परफेक्टली ओके, बस याद करने के लिए आसान तरीका बता रही हूँ। बस मिक्सअप हो जाता है जब आगे पढ़ेंगे बहुत सारी चीजें, ठीक है। तो आपका जो एंडोसाइटोसिस होता है, इट इज फर्दर ऑफ टू टाइप्स। अब एंडोसाइटोसिस के साथ मेक टू एरोज बीनीथ इट, ऐसे दो एरोज बना लीजिएगा। इसमें एक में लिखिएगा पेगोसाइटोसिस और एक में लिखिएगा पिनोसाइटोसिस, ठीक है। थोड़ा सा टेक्निकल बात है, अगर समझ आ जाएगी तो गुड इनफ हो जाएगा, बट ऐसे ही बना लें, पेगोसाइटोसिस और फिनोसाइटोसिस। सो इसको जो है ना थोड़ा सा इन डेप्थ में जाके देखते। एंडोसाइटोसिस है प्रोसेस जिसमें सेल्स जो हैं दे आर गोइंग टू इंटरनलाइज लार्ज मॉलिक्यूल और लार्ज वॉल्यूम ऑफ एनी मटेरियल, ठीक है, फ्रॉम द एक्सटर्नल एनवायरनमेंट। उसमें वी हैव टू मोर कैटेगरीज, पेगोसाइटोसिस एंड पिनोसाइटोसिस। Phagocytosis is usually the involvement of solid particles, and this process is literally known as cell eating ka process, and it is going to involve solid particles, while phagocytosis involves liquid particles. So this process is known as cell drinking process, ठीक है। So in phagocytosis, your cell membrane is going to extend around the particle. It is going to form a vesicle, and that vesicle is going to be known as phagosome, and it is then brought into the cell, okay? But when pinocytosis is concerned, the cell is again going to invaginate. It is again going to form a small vesicle which contains that extracellular fluid, and then the entire material is going to move inside the cell. So basically what I'm trying to say is that be it a solid particle or be it a liquid particle, in endocytosis or in exocytosis, the material is transported in form of vesicles, okay? But when exocytosis is concerned, the cell is going to expel the materials, but again in form of vesicles. So vesicles will contain the substance, and it will then fuse with the cell membrane and it will release the content to the outside environment, because exocytosis involves movement of material outside, while endocytosis involves movement of material inside the cell. Okay. And then here are the steps. So endocytosis, exocytosis, individual 3 mark का question आता है, इसका भी 3 का आ सकता है, इसका भी 3 का आ सकता है, in which you have to write the steps. So endocytosis का first step is engulfment, where cell membrane engulfs the external material. It forms a vesicle. Then vesicle formation में ये होता है that membrane pinches off to form an intracellular vesicle, because the vesicle has to move inside the cell, and then the vesicle fuses with lysosomes. For example, if something has to degrade, अगर कोई waste material आ रहा है and that waste material has to degrade, then that vesicle is going to fuse with lysosomes. Why? Because lysosomes has hydrolytic enzymes, ठीक है, hydrolytic enzymes उसको break down करवाते हैं. Yes, Minha.
मिस ये फॉरेन सेल्स के लिए भी है?
Yeah, this is for foreign cells. Foreign cells में phagocytosis होगा, ठीक है। पैगोसाइटोसिस का पूरा कॉन्सेप्ट हम इम्यूनिटी में दोबारा रिविजिट करते हैं, इसलिए अभी सुपरफिशियली हम एंडोसाइटोसिस को देखते हैं, बट दिस इज एग्जैक्टली गोइंग टू बी द सेम फॉर पैगोसाइटोसिस। तो यस एग्जैक्टली, अगर फेगोसाइटोसिस है उसमें फॉरेन मटेरियल होगा तो इट हैज टू फ्यूज विद लाइसोसोम्स, ताकि लाइसोसोम्स उसका पूरा ब्रेक डाउन करा सके, ठीक है। तो ये फेगोसाइटोसिस में भी यही होता है कि अगर डिग्रेड करना है, एक फॉरेन मटेरियल है तो लाइसोसोम्स के साथ फ्यूज होगा, उसको डिग्रेडेशन हो जाएगी। सिर्फ डिग्रेडेशन के लिए नहीं होता, कभी कभी चीजों को प्रोसेस करने के लिए भी होता है, ठीक है। एंड देन फॉर एक्सोसाइटोसिस, वेसिकल्स जिनमें सब्सटेंसेस होते हैं दैट फॉर्म इनसाइड द सेल, जो सेल से बाहर ट्रांसपोर्ट होने की जरूरत होती है, वो चीजें बनती हैं, वो वेसिकल फॉर्मेशन में आती हैं, एंड देन इट मूव्स अलोंग, तो फ्यूज करती है सेल मेम्ब्रेन के साथ, एंड देन देयर कंटेंट इज रिलीज्ड आउटसाइड द सेल्स, ठीक है। सो दिस इज इट, and with that being said, we have concluded our chapter number 4. We planned to do our past papers today, but few of you did not listen to the recording, so koi baat nahi. We'll do the past papers tomorrow, and one announcement is that next Saturday you will have the test of cells, chapter 1 and chapter 4, cell membrane, okay? So on next Saturday, try to attend the class in a live manner, because test लेना है मैंने, तो दोनों chapters को अच्छे से तैयार कर लीजिएगा. I also post the homework. I did not give the homework last week because मैंने क्लास ही नहीं ली थी, तो homework क्यों देती, बट मैंने अब क्लास ले लिया, तो अब मैंने होमवर्क भी देना है, एंड आप लोग तैयारी भी स्टार्ट करते हैं सेल्स एंड सेल मेम्ब्रेन की. ऑल राइट, Do we have any questions?
No.
मैम
Clear है
No,
सारा?
Yes.
All right, perfect. ठीक है जी. Take care and jazakallah khair for joining. Assalamu